Good day to everyone joining us, and welcome to today's Eurofins Panlabs webinar. Today's talk is entitled Oncopanel, 3D Tumor Spheroids and Long-Term Assay for Epigenetic Therapeutics. My name is Mike Futala, and I'll be your host today. Today's webinar will run for approximately 60 minutes and includes a question and answer session. This webinar is designed to be interactive, so please feel free to submit your questions and comments for our speaker throughout the presentation using the question chat box, and we will attend to your comments and questions during the Q&A session. This chat box is located in the control panel on the right side of your screen, and if you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode, and the presentation slides will advance automatically. Please also note that this event will be recorded and made available to you for future download. Okay, a brief history of Eurofins Panlabs Incorporated. Eurofins is a global CRO specializing in discovery pharmacology testing services for over 40 years. Eurofins Panlabs' mission is to provide pharmacological testing services that predict clinical effects. Their scientific portfolio consists of over 1,800 assays, ranging from molecular assays to cell-based models through proof of concept in vivo activity determination. Now I'd like to introduce the speaker for today's event. He's Dr. Jamil Shah. Dr. Shah is responsible for logistics, oversight, and development of the Oncopanel platform. He is an experienced drug discovery scientist in the pharmaceutical industry and has expertise in the area of cell biology and oncology with a proven track record of success and innovation. Dr. Shah has a PhD in cellular and molecular physiology from the Pennsylvania State University College of Medicine. He completed postdoctoral fellowship at the Salk Institute for Biological Studies in the field of cell growth control and signal transduction. He has received numerous awards, including several issued patents, and has authored 19 published peer-reviewed research articles. So now I'd like to hand over the presentation to Dr. Shah. Please remember to chat in your questions in the chat box, and we will address them in the question and answer session that follows the presentation. Okay, Dr. Shah.
Okay, thanks, Mike. Okay, so good morning, everyone. I've essentially broken this talk into 3 parts. The first part, I'll provide an overview of the Oncopanel line of service. The second part, I'll discuss Oncopanel LT. That's our long-term cell line profiling offering. And the third part will be a discussion about Oncopanel 3D, which is our new 3-dimensional cell culture assay. Okay, so if there's only one message to be taken away today, it's that Oncopanel is a predictive biomarker discovery platform, and perhaps this is easier to appreciate by first considering that cancer results from the acquisition of a particular kind of genomic alteration. These are sometimes referred to as driver mutations, and these provide cancer cells a selective advantage which allows them to do things like replicate beyond their architectural constraints and resist the body's safeguards against inappropriate proliferation. So these phenomena are now familiar to most of us and have been cataloged very nicely by Hanahan and Weinberg and termed the hallmarks of cancer. This was all published in a review now over a decade ago. However, this is not the whole story, and there's actually a flip side to this coin, and that is that the driver mutations themselves are also addictive to the cancer cell. So what this means is that by inhibiting these cancer drivers, you can produce a therapy that's both effective to halt the cancer's progression and is also targeted to the specific molecular alterations that drive an individual's cancer. So cancer drivers themselves represent valuable biomarkers to predict response to therapy. I'd like you to consider a couple of clinical trial scenarios. In the first case, the first clinical trial scenario, the predictive biomarker is not known. And because there's a poor understanding of who's likely to respond to therapy, you have to enroll a large cohort of patients at a high trial cost. The individual probability of success is low, again, because we have little power to predict who's likely to benefit. And the benefit to the individual patient, that is, the mitigation of their disease, is also relatively low. So you can contrast this with a scenario in which predictive biomarkers are understood. And in this scenario, you can intentionally exclude patients not likely to benefit from therapy and enrich your patient cohort with those individuals likely to benefit based on the presence of a predictive biomarker. So in this case, the overall trial size is smaller, and you incur reduced cost in enrolling this smaller cohort of patients. Probability of success for the individual patient is higher, and the overall benefit, that is the therapeutic response for the patient, is also likely to be higher. And here's a few examples of successfully implemented predictive biomarkers. So one example is using HER2 expression to predict response to the anti-HER2 antibody trastuzumab, EGFR mutations to predict response to the EGFR inhibitors gefitinib and erlotinib, and actually KRAS mutation to predict resistance to the anti-EGFR therapies cetuximab and panitumumab, so that patients enriched in these trials were wild type with respect to KRAS. So for those who are new to Oncopanel, I wanted to spend a few minutes and tell you about what it is exactly. So it's one of the largest commercial profiling panels of cancer cell lines. The current number of cancer cell lines represented is 240, although there are plans for significant expansion of the panel over the course of the next year. We currently have a very broad representation of cancer types and subtypes, and we have extensive genomic characterization of all 240 cell lines, wherein 100% have mRNA expression coverage. This comes from both internal and external sources. And we have mutation and DNA copy number data that comes from chiefly two public repositories. One is the COSMIC database from the Sanger Institute, and the other is the Cancer Cell Line Encyclopedia from the Broad Institute. Most cell lines are in license from ATCC. We do obtain a few lines from other banks like DSMZ, Riken, et cetera. We have a large cell line bank that ensures consistent performance year to year, so a client can expect data generated in 2009 to be similar to data generated today. One of the things that we take great pride in is the meticulous tracking of every cell line's historical performance. Again, this sort of goes back to Oncopanel's inception. Doubling times are recorded at each passage after they've been thawed and within every assay. So given that we've run Oncopanel several times per year, this is an extensive recording of historical performance. The seeding densities are specific for each cell line, and they're based on both the doubling time of that cell line and the goal of reaching 80% confluency at the end of the assay. This is independent of the assay period, whether it's a 24-hour assay or a 10-day assay. We still have 80% confluency as our target. And we assay all cell lines at low passage. All cell lines are passage less than 3 times at the time of the assay. And this ensures that they don't drift genetically, and that therefore allows us to relate the drug response to genomic information that may have been provided in the public domain. So what makes Oncopanel unique? There are a number of cell line profiling companies out there. One of our distinguishing characteristics is that we're a high-content platform. Instead of performing a homogeneous mix and read assay, we can instead measure multiple parameters from the same well, and these parameters can be tailored and customized to detect essentially anything the client is interested in. Our standard offering is listed here, and that is we measure cell count by staining the nuclei with DAPI and then counting the number of cells remaining in the well. And for the cell count dose response curve, we report a number of different drug response parameters such as EC50, IC50, GI50, and the integrated area above the dose response curve or activity area. We also measure apoptosis by detecting the cleaved active form of caspase-3. This represents the committed step in apoptosis. And then additionally, we report cell cycle information by detecting and quantifying the levels of phosphorylation of histone H3 on serine 10. So this is a marker of the G2M transition. Additionally, we also report doubling times for each cell line within the assay. So you can understand sort of how growth rate may relate to overall drug response. So as I sort of alluded to a moment ago, this is a highly flexible and customizable assay so that, for example, clients who may be more interested in measuring a predictive biomarker, a pharmacodynamic biomarker, instead of one of our standard endpoints, we can swap these out and allow reporting of that parameter instead. We also have validated a number of different assay incubation periods. We've done everything essentially from 1 to 10-day incubations for clients, and we also do some more complicated procedures like drug washouts and things of that sort, compound readdition, etc. And finally, one of the things that we've now rolled into this whole package is an integration of predictive biomarker analysis. We do a univariate genomic analysis as well as a pathway analysis as part of this service, and this is to help clients understand which genomic biomarkers may be related to drug response. So here's the Oncopanel workflow. Again, we start with a collection of histologically diverse cancer cell lines, and we have so far really tested anything that can be developed from a therapeutic standpoint. We've tested nanoparticles, peptides, recombinant enzymes, antibody-drug conjugates, and of course small molecules and biologic therapeutics as well. We also do drug-drug combinations where one can mix and match different therapeutics to look at their combined activity. We use a state-of-the-art imaging platform. So we have 2 Image-Express Micro XLs, which are regarded as state-of-the-art. We have the ability to image at 4x, 10x, or 20x magnification, and we can do this in sort of high throughput fashion. And we can image and quantify signals from 4 channels simultaneously. So our data analysis in QC is extremely stringent, and I'll list some of our quality control criteria here. So on every plate for each cell line, we run a reference inhibitor, staurosporin, on every plate. And the performance of staurosporin must be within 3-fold of the historical staurosporin average, both with respect to EC50 and IC50. So the data need to be very consistent. Furthermore, we require that the doublings for a given cell line be within 50% of its historical average. Again, this sort of ensures that the cells are growing and behaving as we'd expect. Then, as I mentioned, the last step is to identify and prioritize predictive biomarkers through the genomic analysis. This is an example of what some of the data actually look like. So on the bottom, you have sort of a composite fluorescent image looking at 3 different fluorophores simultaneously. What's shown in green is apoptosis, cells undergoing apoptosis. What's shown in blue are just individual nuclei, and shown in pink are those that are positive for the phosphohistone H3 serine 10 marker of G2M. And the corresponding dose response curves sort of plotted according to the same color scheme above. So what a client can expect then is data on all 240 cell lines from multiple parameters from each of these drug response measurements that we record. And an example is shown here. And using these waterfall plots, we could use any one of these drug response parameters as the basis for a genomic analysis. For example, the question might be, what are the genomic biomarkers associated with significant induction of apoptosis, in which case we could provide information there. As I mentioned, we also do drug-drug combination testing. This is just one example where we've computed excess over Bliss independence, and I've highlighted some of the cell lines that show synergistic activity of these two drugs. And from this data, we can do things like compute a Bliss sum, which is the sum of Bliss excess measurements over the entire dose-response curve, and generate a waterfall plot analogous to the one you saw before, and then perform a genomic analysis on these data, again with questions. Questions might be, what are the genomic traits associated with synergistic activity of these two drugs? So just a slide on data quality, and I should preface the description of this slide by saying that these data were actually collected on our previous imaging platform, which I will show you is even less robust than the current imaging platform that we use. On the top are the cell count vehicle CVs. One of our QC criteria is that these be less than 0.3 for each cell line, otherwise we'll reject the cell line and repeat the experiment. Oncopanel typically is run over an 8 or 9 week schedule with about 30 cell lines per week. So I'm showing you the results from 3 successive Oncopanel runs. Each circle represents a different cell line. Each color represents a different Oncopanel run. You can see that overall the variability in this assay is quite low and consistently that way. On the bottom, this is another level of granularity shown. Again, color coding is by different Oncopanel runs, so 3 successive runs are shown. And what I wanted to point out was that actually 90% of the vehicle CVs are less than 0.1. So the majority of the data is extremely tight. And again, this is not as good as the data quality using the current imaging platform, and I'll show you another slide that speaks to that. So the newer imaging platform that we've upgraded to takes advantage of a technology that allows whole well imaging at 4x magnification. And because you can visualize more of the well, the data quality is actually significantly improved. And that's illustrated on this slide. So each plot represents one of our standard drug response parameters, cell count on the top, apoptosis in the middle, mitosis or G2M in the bottom. Each bar represents a different cell line, and the blue are data that were collected on the old imaging platform, the NCell 1000, our first generation platform. The pink represents data collected on our newer Image Express platform, and you can see the average vehicle CVs are much lower, shown in pink, compared to the original platform, shown in blue, for all 3 parameters. So we've significantly improved data quality. So one of the reasons that looking at doublings within the assay is important is that in some cases, when the mechanism of action, for example, is poorly understood, this can provide some useful information in terms of how the drug is behaving and what drives drug behavior. So if you take, for example, the 3 reference compounds that we've tested in Oncopanel, each line, each curve represents the dose response curve for a different cell line, and the color coding is by the number of doublings within the assay. So red indicates a cell line that doubles quickly, green indicates a cell line that doubles slowly. And what you can see quite readily by looking at these plots is that there are a number of therapeutics whose drug response is tightly correlated with growth rate. And this is in contrast with other inhibitors like CI-1040 or Lotinib, which are more targeted in nature and whose drug response is not correlated with cell growth rate. Since Oncopanel began, we've sort of always run a set of reference compounds, and we continue to do so. And in fact, we will happily accept suggestions from clients or prospective clients about which therapeutics we could run as standards as reference agents. And these data are also sold. So if somebody's interested in the drug response for a reference inhibitor, we can sell that as well. But what we can do is take a client's compound and cluster the drug response against this reference collection to try to provide some insight into how their drug behaves in relation to other drugs with known mechanisms of action. One of the other things that sort of is borne out in this particular slide is you can see that agents, chemically distinct agents with a shared mechanism of action cluster quite similarly. And this also speaks to the quality of the data. We wouldn't see this clustering similarity if the data quality was poor. Okay, so that's an overview of Oncopanel. So what I think I'm going to do now is switch gears and tell you about Oncopanel LT, LT for long-term. And the field of epigenetics over the last few years has grown quite rapidly, and there are a number of pharmaceutical companies that are actively developing therapeutics in this area for the treatment of cancer, both in terms of sort of homegrown initiatives as well as in strategic partnerships with other epigenetics companies. One of the challenges, however, that we face developing drugs of this sort is that we need to, in some cases, measure a delayed response of that drug. And in some cases, this can take several days to fully manifest. So this can be due, in fact, to drug targets that must first modulate histone or DNA modifications, which in turn affect the packaging of DNA into nucleosomes, and then in turn again affect the expression of genes in this region of DNA. So to try to address this issue, we set out to develop a method that would allow cancer cell line profiling with incubations as long as 10 days. And this is to identify both sensitive and resistant cell lines and cancer types, as well as to identify the genomic biomarkers that predict drug response. An example of this is shown here. So what we've done is taken advantage of two inhibitors of the histone methyltransferase EZH2. The first is GSK343, and the second is ONC1999. And we've tested these inhibitors in a cell line that bears the EZH2 mutation called Pfeiffer. So these are a comparison of a 10-day and 3-day assay result. And you can see for both EZH2 inhibitors, there's sort of little activity detected in 3 days, whereas the potency of these compounds is significantly improved or significantly shifted to the left with longer incubation. And on the bottom, this is contrasted with staurosporin, which sort of has no particular genomic predisposition, where the 3-day and 10-day assay results are the same. So when we began to sort of set out to validate this this 10-day assay, we sort of decided that our treatment protocol was going to need to look something like this. So on day 0, we seed the cells. Day 1 would be compound treatment, and like we do for Oncopanel, on day 1 we also process a second plate at the time of compound treatment that represents the number of cells at the beginning of the assay. We use this both to compute the doubling time and to compute the GI50 value, the growth inhibition 50. And then we incubate for 6 days continuously. We knew we were going to have to seed these cells at relatively low density compared to our 3-day assay, and therefore we also knew that the cells would sort of not deplete their nutrients from the growth medium during the first 6 days or so. On day 7, we change the medium, and then we add the drug a second time, so drug readdition on day 7, incubate for a further 4 days, and then we fix, stain, and image the plates on day 11 for a total of 10 days of incubation. So for our validation experiments, we also wanted to work with cell lines that covered a range of growth rates. So we took extremes in terms of doublings, extreme on the high end and extreme on the low end, as well as cell lines that grew at a mixture of adherent and non-adherent cell lines, and cell lines whose doubling times were more or less in the middle. The first question that we wanted to ask ourselves is how well we could seed cells at low density. And so this is simply a serial dilution of, at the time of cell line seeding, to try to understand the accuracy with which we can seed cells. And what you can see over this sort of range of cell lines of different growth properties, that is adherent versus non-adherent, we can accurately seed these cells down to about 15 cells per well. And you can see in many cases the R-square value of this is 1.0. So this gave us some confidence that seeding the cells at low density was not going to be an issue for us. The other question was whether or not we'd be required to split cells over the course of the assay. And speaking with some of our clients, we had learned that some of the experiments performed in-house required that they actually trypsinize and reseed cells, or for non-adherent cells, simply split them over the course of the assay. Knowing what we did about Oncopanel, we were concerned that this might introduce some variability, but we wanted to directly test this. So what I've plotted in the bottom right plot, on the y-axis is CV. Again, as a reference, the horizontal dashed line indicates our sort of quality control criterion in terms of what's acceptable. Each cell line is plotted as a different color, and each symbol represents a different seeding density. So we looked at vehicle CVs over the course of multiple days during the 10-day incubation. And what you can see is if we don't split the cells, the vehicle CVs are essentially within acceptable limits, although for a couple cell lines at the very low densities, the CVs start to creep up just above what's typically considered our acceptance criterion. When you contrast this with splitting the cells, so if you split the cells and you replate them, the CVs are completely unacceptable. So we knew that this was not going to be an option for us if we were to offer this assay and still adhere to the sort of quality that our clients have been accustomed to. So we'd convinced ourselves that we could seed the cells at low density. And the next question we wanted to ask was whether or not we'd be able to predict what the optimal seeding density is for each of the 240 cell lines to try to achieve 80% confluency at the end of the assay. So we then sort of returned to our historical data and simply extrapolated sort of our growth out to any given incubation period, 5 days or 10 days, and then back calculated to determine what the optimal seeding density was. So this assumed that we would observe logarithmic growth for all 240 cell lines. And if we did observe logarithmic growth, the historical data should allow us to predict what the right density should be. And to determine that, we plotted the ratio of the predicted to measured doubling. And as long as these were in 50% of a value of 1, a ratio of 1, we would consider these data to be acceptable. So we first did this for our 5-day incubation, and we were encouraged to realize that we could, in fact, predict doublings accurately. And in 96% of the cases, these predictions held true. So then we went back to our 10-day validation, and we wanted to ask directly, what was the growth exponential for the cell lines that we happen to be testing over a range of doubling times at their optimal seeding density? This, again, is based on what we calculated. So what you can see here is that the growth, for the most part, is exponential for the cell lines tested. The exception perhaps being HCT116. Again, this was our fastest cell line, and where the estimated seeding density was actually less than 15 cells per well. So what you see is sort of logarithmic growth for about the first 7 or 8 days, and then sort of a slowing in growth in the last couple days. And we do, so for some of the fast-growing cell lines, we do expect this is going to be their pattern of growth. But based on these calculations, we also estimate that about 83% of the cell lines are going to show logarithmic growth over our 10-day assay, whereas maybe around 15% are going to show the slowing during the last phase. So then we, now that the validation was complete, we began to put test cell lines, all 240 cell lines, under our 10-day incubation conditions. And then we sort of revisited this question about how well we could predict doubling times. And in this case, over the 10-day incubation with our current protocol, we would accurately predict the doublings in about 3/4 of the cases. This meant that 76% of the 240 cell lines grew logarithmically because the doublings were predictable. In contrast, we had about 24% of the cell lines that grew a little slower than predicted. So when we looked carefully at these cell lines, one of the things that we noticed was they sort of didn't really grow well at all. And one of the things that one of the reasons we think this is the case is that some cell lines do require some paracrine mechanisms to keep them happy during growth. So what we've done with each of these cell lines is we've iteratively tested and checked the seeding densities when we gradually increase them. And we increase them to a point until we reach 80% confluency at the end of the 10-day incubation. So you can see the before and after seeding density adjustment, and that now we've achieved our target confluency. So after having gone through this entire process, when you look at the growth overall of each cell line within the collection of 240, the cells actually grow quite well overall, with a median doubling time of about 5.6 doublings in 10 days and a maximum doubling time of 9 doublings in some cases. So this was all well and good, but the real question, to really validate this profiling service, we have to convince ourselves that the form in which the profiling is offered is sufficient to identify the right genomic biomarkers. So again, we returned to sort of our case study, which was the EZH2 inhibitor GSK343. We tested this inhibitor in all 240 cell lines, and what I've done here is I've binned the EC50 values by potency. So these are binned into half-log buckets. In the top left are the cell line dose response curves that are the most sensitive, and this moves to increasing levels of resistance. So what I've highlighted too are the cell lines shown in pink, which contain the EZH2 mutation. And when you plot these data as a waterfall plot, you can see that the EZH2 mutant cell lines are those that tend to be the most sensitive to this particular drug. So we then categorized cell lines, classified them as either sensitive or resistant, and then performed our genomic analysis. And what we found was, again, quite encouraging. So we found not only EZH2 mutation, but a number of other genomic features related to epigenetic mechanisms as significant based on our significance calling. And all of these were found, in this case, were found to be sensitive to predictive of sensitivity to the EZH2 inhibitor GSK343. So a number of these epigenetic modulators are shown here, as well as proteins known to directly interact with EZH2. So this was encouraging. It meant that we had developed something that sort of served its purpose. I'm not going to go through all of the genomic analysis. That's sort of a topic in and of itself, but this was one other, this is kind of one other piece of data that clients can expect as part of their genomics analysis report, and that's a look at the amino acid positions of the mutations that were found to be significantly associated with drug response, in this case sensitivity. So you can see in this case that the EZH2 mutations generally fall into two positions that are known to be sort of oncogenic. Okay, so that that summarizes the Oncopanel LT offering, and now I'm going to sort of wrap up with the discussion of Oncopanel 3D. So one of the caveats that's associated with traditional two-dimensional cell culture is that cells in 2 dimensions don't experience the same nutrient metabolite, oxygen, interstitial pressure gradients that are experienced in sort of 3-dimensional models such as tumor xenografts or autochthonous tumor models. So, and this may explain some of the discrepancies in efficacy that one can see when you compare these 2 models. So for us to culture cells 3-dimensionally, we chose to use cell-able oncology plates. These are 384 well plates into which each well is arrayed a matrix of 250 collagen-coated spots, so that the cells will specifically adhere to these spots and form 3-dimensional aggregates. They typically range in size anywhere from 100 to 300 micrometers in diameter, and they grow in a nice organized way. And this was, you know, in part because there were some unique applications related to high content imaging that we could deploy when we utilize these plates. One of the in vivo characteristics that we thought was going to be important to try to phenocopy was the presence of hypoxia in the center of the 3-dimensional aggregate or spheroid. So we developed a method to visualize hypoxia in each object, and we used two distinct dyes that are both live cell dyes. First, we used a DNA stain, HEST, to determine where all the cells are. And then we use a dye called LOX1, whose fluorescence is quenched in the presence of oxygen to visualize objects whose interior is hypoxic. So using these 2 fluorophores together, we then apply a spheroid calling strategy that allows us to compute the number of cells growing in spheroids. So an example, you know, if you just focus in on a portion of the well, you can see here that only 2 of the spots actually are growing spheroids, whereas the other 2 have cells that are sort of either individual cells or cells growing in what's probably a monolayer. So we would sort of exclude that from our calculation. And we use that as sort of a quality control step. So what we wanted to do initially, you know, you can imagine that the validation of 240 cell lines is not a trivial task, but we had to come up with some method to be able to sort of determine in a cell line by cell line way what is the growth period that's required to establish these spheroids before drug treatment. And again, spheroids or aggregates are defined by the presence of hypoxia in the center. So what we did is we grew each cell line for a period of up to 2 weeks. We stained with our 2 live cell dyes, and then we imaged to understand when spheroid formation occurred. So on the bottom left-hand plot, you can see spheroid growth over time. And so we have, again, here a range of cell lines based on growth rates, BT474, which grows slowly, and contrast that maybe with HCT116, which grows quickly. And you can see spheroid formation occurs at different periods. It occurs really only after 2 weeks for BT474, and occurs relatively quickly, as soon as 6 days, 3 to 6 days, maybe for HCT116. And some representative bright field and fluorescent images are shown on the right. Again, just showing that hypoxia appears according to the same time schedule. So not surprising then, the time frame required for a cell line to form a spheroid is related to its growth rate. And here growth rate is just plotted as the number of doublings in just 3 days. Again, knowing that we can sort of extrapolate this value out. Not a particularly surprising result. So as we sort of systematically marched through each cell line, we noticed that the growth properties were distinctive and essentially fell into one of 3 buckets. We had the first bucket that we classified as non-aggregate. This is where cell lines didn't grow as multicellular aggregates at all, but rather as individual cells. You can also see we're not detecting any hypoxia, as we would expect. The second we classified as loose aggregates. These were spheroids that did form a multicellular aggregate and also displayed a hypoxic core, but sort of lost this nice organization that one would expect in a true spheroid. And then on the bottom are those cell lines that grow as what we would classify as a spheroid. Again, these form these nicely organized 3-dimensional objects, and again, hypoxic in the center. So after sort of characterizing all 240 cell lines, we were left with about 47 that formed loose aggregates, 97 that formed true spheroids, and about 94 which in the 2-week incubation didn't really form spheroids. We classified those as non-aggregates. And then if you break this out further by cancer type, a couple things maybe to highlight are that in general, hematologic malignancies don't form spheroids or loose aggregates particularly well. You might contrast this with cell lines from the large intestine, which form some 3-dimensional aggregates as a preference. So one of the other interesting questions that we could ask, given we had a genomics analysis capability, was what are the genomic features that are required for spheroid formation? So we perform this assay by binning cell lines into either spheroids or loose aggregates, and then using mRNA expression, mutation, and DNA copy number as part of our genomic analysis to try to highlight those significant genomic features associated with either aggregate type. And then we took those significant genomic features and performed a pathway analysis to try to understand if there are certain biological mechanisms at work that would drive spheroid formation. Initially, one of the sort of interesting things that was also sort of intellectually satisfying in a way was that loose aggregates tended to be derived from metastatic cancer rather than primary tumors. And again, this loss of organization has been appreciated for some time to be sort of a force at work during metastasis. So this was one sort of early encouraging observation. So when we looked at the pattern of differential expression, and what we were using a 5% false discovery rate as a cutoff for significant differentially expressed genes, and we looked at the genes that were upregulated during spheroid formation, and we found a number of highly differentially expressed pathways that were related to cell-cell junction formation. And this sort of took on a number of different flavors. It wasn't just this, but also actin dynamics, processes that regulated sort of the dynamic state of actin in the cell, as well as RAC and Rho signaling were highly enriched. And when you look sort of closer at sort of this, you know, these cell-cell junction pathways really fall into 3 main categories. Those associated with the structural components that form desmosomes, tight junctions, and adherence junctions. So collectively, these things were working together to enable spheroid formation. And when you actually sort of look overall at all of the genes that were considered upregulated during spheroid formation, almost 70% of those genes fell into this larger bucket of either cell-cell junction or actin dynamics. So that's really sort of a striking result, but also makes a lot of biological sense because we knew this organization, cell organization, was going to be important. So in terms of what we offer in terms of a 3D testing protocol, one of the offerings that we think is actually going to be most useful is a comparison of 3D drug response to 2D drug response. So I've outlined sort of our procedure to test drugs in our 3D model. At the time of drug addition, we perform this quality control step to really ensure that the majority of the cells are in spheroids at the time of drug testing. The drug testing is for 7 days, 7-day incubation, and then we process all the spheroids in the well using cell titer glow 3D, which is a new reagent from Promega, specifically designed to lyse 3-dimensional spheroids. And at the same time, in parallel, under the sort of the same incubation period, we perform an assay in 2D. So this is a monolayer experiment, and then this allows clients to relate the drug responses under these two different culture conditions. So here's an example of 4 compounds with different mechanisms tested in 2 different cell lines. The 2D result is shown in green, and the 3D result is shown in blue. You can see that in some cases there's a significant shift in potency depending on the compound and depending on the cell line. And generally, we see sort of weaker activity in 3 dimensions. Again, we think this is associated with the poorer ability of drugs to penetrate into the tumors, into the 3-dimensional aggregates or spheroids. We do have some examples, however, where the drug response is actually flip-flopped. And if you consider the result for selenomycin, which is a mitochondrial respiration inhibitor, for example, this is actually more active in 3D culture than 2D. And we think this probably has something to do with the metabolic activity of the cells when they're either grown under hypoxic conditions or otherwise grown restricted from nutrients within the spheroid interior. So for people considering Oncopanel, one thing that's important to appreciate is that this is run as a consortium, and it's run 3 to 4 times every year at regular scheduled intervals. Our next run is actually June the 16th. The turnaround time for the Oncopanel service is 12 weeks, and we run 30 to 35 cell lines per week on average, and there are 2 data deliveries over the course of that 12-week period, one after 6 weeks and then one after 12 weeks. So I'm going to just sort of recap the highlights. First, for Oncopanel LT, we have 240 cell lines that we've validated for long-term culture. We perform incubations of up to 10 days with drug. Again, this is with a medium and compound readdition at day 7. And we also, however, perform incubations at shorter periods. So for example, we have clients currently where we're performing a 6-day incubation. We have some we're performing a 4-day incubation. So essentially, clients can come to us and we can set up an assay, an assay that we've already validated for any incubation period up to 10 days. The genomic analysis is included free of charge if all 240 cell lines are profiled. For smaller profiling services, we do charge a small fee for the genomic analysis on a per compound, per parameter basis. And this service is really ideal to profile drugs that have a protracted or delayed mechanism of action, where, for example, a shorter 3-day assay is not suitable. Oncopanel 3D, we have 144 validated 3-dimensional cell culture models. Again, about 97 spheroids and 47 loose aggregates. Clients can cherry pick for many of these models. The same cherry picking is also an option for Oncopanel LT. Our quality control involves hypoxia-positive spheroid culling to ensure that the cells at the time of treatment are truly hypoxic in the center. And We also offer the ability to profile 2 and 3D simultaneously to allow comparison of drug response in the same cell lines based on the 2- or 3-dimensional growth conditions. So this is sort of ideal to compare molecules with improved tumor penetration or with otherwise improved activity in 3D. For example, it might be this compound, you have a compound that was designed to synergize with hypoxia. Maybe you'd see more activity in 3-dimensional culture here. I should point out that due to the sort of differences in the ability of drugs to penetrate tumors, and that difference is cell line specific, we actually don't think that 3D is suitable for biomarker discovery. So the 3D cell culture offering is probably better going to enable sort of a comparison of compound effects rather than identify genomic biomarkers. And then finally, the Oncopanel 3D service is going to be rolled out next month. And I was told to mention that there's a promotion that's currently going on for new Oncopanel clients. And you can sort of go to the website at Eurofins-Panlabs.com and sort of view the promotion. So with that, I think we're going to try to take a few questions. So we're going to take a moment and review the questions that have been submitted, and then we'll be back online and try to address some of them. Okay, so I can try to take a few questions here. So the first question is, in the Oncopanel cell lines, do you have them classified according to RAS, P53, et cetera? And what I can say to this is that we have genomic information covering about 1,600 genes for all 240 cell lines. And so what we do with clients is if they are interested in a particular, the mutational status of a particular gene or a particular collection of genes, then we sort of review the genomic data for them and provide them a customized list of cell lines associated with the corresponding genomic characteristics. And this is often done sort of in the beginning and helps clients, cherry pick which cell lines they'd like to test if there is a sort of mutational background that's of interest. Second, can we use gene expression, copy number, mutational data of Oncopanel 240 cell lines growing in spheroid culture, not growing in 2D? The short answer is yes, you could, but I think maybe the bigger question is, would you want to? So as I mentioned before, although the genomic information does exist and things like mutation and copy number data are going to be fixed, that is, they're not going to be changing over the course of either the culture condition or the assay incubation, the confounding factor here is going to be that depending on the architecture, the three-dimensional architecture for each spheroid model, drugs are going to have a different ability to penetrate into the interior and therefore kill the spheroid. And so this tumor penetration phenomenon is always going to confound the biomarker analysis if purely done in 3D. So another question here is, why do only some spots on cell-able plates form spheroids? What we have tried to do is seed the cells in such a way that we have not all of the spots occupied by spheroids. And we do this by seeding the cells at a relatively low density, which is usually less than 1 cell per spot. And that's why you see some spots occupied by spheroids and some that are not occupied by spheroids. Fourth question, does the expression of RNA change depending on whether the cells are in 2D or 3D culture? So I'm sure the answer to that question is yes. I'm sure that you know, when cells are organized in 3-dimensional culture, what they experience is different. You know, simply changing the nutrient concentration in the center is going to change the expression of genes within the interior of the spheroid. So I think absolutely you're going to expect that. You're also going to have heterogeneity in expression across a single spheroid, more than likely. So measuring RNA expression at the end of the assay is not something that we do, but we might be able to provide clients with samples that they could then submit for RNA expression profiling later if that was of interest. Let's see, it looks like we might have one more question here. Does RNA expression change over the course of the experiment? For example, after a medium change at 7 days? You know, again, I would, I'm sure that there are some gene expression changes that will sort of change over time, particularly as it relates to changes in nutrient status within the culture. As nutrients are depleted, you know, there will be gene expression changes that reflect that. On the other hand, you know, I think what we've learned is that over the course of these experiments, changes that really relate to a cell's intrinsic response to drug probably doesn't change a lot, because we you know, identify things that relate to drug response. We identify gene expression changes that relate to drug response after the 7-day incubation, and these gene expression changes are sort of defined before the experiment even begins. So, so while there are probably some gene expression changes that take place, there are probably others that do not.
Great. Thank you, Dr. Shaw, for those very interesting and informative presentation and all your answers to the questions. And now we've reached the end of the question and answer portion of the webinar. If you do have further questions, please direct them to Dr. Shaw's email address showing on your screen. And thank you, everyone, for participating in today's webinar, and you will be receiving a follow-up email from Eurofins Panlabs with access to the recorded archive for this event. Also, as you leave, a survey window will be popping up on your screen, and your presentation participation is appreciated, as it will help improve our future webinars. So now please join us in thanking our speaker, Dr. Shaw. We hope that you found this conference informative, and have a great day, everyone. Thank you.